Welcome back, everyone. If we can have our first slide. So I'm delighted to start our final session on HIV prevention and cure. We have 4 fantastic speakers. We will have 3 plenary talks, followed by a clinical case presentation and then a roundtable. We will also stay very much on time, and in respect for everyone, we'll try to finish very close to an hour and a half, a bit more. So it's my pleasure to introduce our first speaker. Steve Deeks is Professor of Medicine in Residence at the University of California, San Francisco, and faculty member in the Division of HIV, Infectious Disease, and Global Medicine at the Zuckerberg San Francisco General Hospital. Dr. Deeks is one of the principal investigators of DARE, the Delaney AIDS Research Enterprise, which is an NIH-funded international collaboratory aimed at developing therapeutic interventions to cure HIV. He's the co-chair of the Towards an HIV Cure International Working Group and a former co-chair of the NIH Office of AIDS Research Towards a Cure Planning Group. Additionally, Steve is a member of the board of directors of the UCSF-based AMFER Institute for HIV Cure Research. Steve has been a pioneer in HIV research for the last 30 years, generating novel and out-of-the-box concepts that later become breakthrough discoveries. He has over 500 publications covering a wide range of topics, and there's really no one better to speak to us on HIV Cure, and I'm delighted to hand it over to Steve.
Well, everybody, what I'm going to do over the next 15 to 17 minutes or so is to sort of just do a big, broad overview of where we are right now in terms of developing an HIV cure. And I'm just sort of going to go through and hit on what I think are sort of the greatest hits in the various different cure trajectories, focusing on what's actually happening in the clinic. But before I get there, well, here are my disclosures. I've had some research support from a couple companies, and I do consult with others on cure-related activities. But before I sort of dig into deeply in terms of these various different trajectories, I need to talk about what's really kind of one of the great unanswered questions is why do we actually need a cure? And it's important to talk about this because we need to figure out what the unmet needs are with current HIV therapeutics so that the cure that we develop will address those unmet needs. And so what exactly is the reason why we're doing a cure? Well, I think most of us realize that treatment is incredibly effective, but there are issues associated with long-term treatment, long-term HIV infection, including stigma, discrimination. There's also this potential for long-term toxicity from therapy, weight gain, comorbidities, and so forth. None of this has been clearly teased out in terms of what drugs are causing what, but there is a general concept that long-term treatment addresses most, but not all, of the issues associated with HIV infection. So those are the direct costs on an individual level that we need to fix with a cure. But probably the main driving reason, I think, in my opinion, to develop a cure is the fact that lifelong treatment, giving people antiretroviral therapies on a daily basis or even monthly with injections, is expensive. It's hard to scale. And despite the fact that there's been this massive global investment in terms of providing treatment for everybody across the world, less than 50% of the population now is on therapy. So we need to come up with an approach that will be safe, effective, scalable. And I will say right up front that the various different cures that we're working on right now will not achieve any of that. Right now in cure research is what I think of doing multiple shots on goal. We got 5 or 6 different broadly defined approaches that we can take. All are slowly being moved into the clinic. They're shown here. None of these by themselves, the way they look right now, are impactful. But the hope is over the next several years that we'll begin to develop a sense of which of these various different trajectories is going to be most plausible, build on that, and then ultimately come up with a cure which will be scalable, effective, and basically address all the unmet needs. That's the long-term goal. And the short-term goal, of course, is to just find proof of concept that there are viable pathways there. So when I think of how to go about curing HIV, I think of sort of these five broad areas. One is starting therapy very early. I'll talk about that quickly. One is the concept of either shock and lock or block and lock, basically manipulating the reservoir. The other is gene therapy. All these are generally designed to cause the reservoir to become quite low or to eliminate completely. On the other hand, we are developing the capacity through a variety of different mechanisms, vaccines, vaccine adjuvants, antibodies, immunotherapy in general, to come up with ways to enhance the control. Fundamentally, these are not siloed. In fact, we're mixing and matching this stuff all the time to come up with a combination of approaches. So let's get going on the various different trajectories. One is early ART. There has been this concept shown mainly in monkeys that if you initiate therapy really early, within the first few days or weeks of infection, you might cure monkeys. But that, unfortunately, has not panned out in people. It is now pretty clear that if we initiate antiretroviral therapy in early HIV infection, in phoebic 1, within weeks of the virus becoming detectable, and in fact, in the case that we have in San Francisco, on the day the virus became detectable, we had a gentleman who started antiretroviral drugs because he was going on PrEP. You basically, with antiretroviral therapy alone, cannot get treatment in early enough to cure people. You can cause the reservoir to be massively reduced, but it by itself is not going to be curative. So treatment early is not curative. But I'm one of the people who believes that early treatment is going to be very beneficial, not in terms of a true cure, but in terms of developing the so-called post-treatment controller phenomenon. And what am I talking about here? So going back several years now, the French and the Visconti cohort identified, and others have since confirmed, that if you initiate antiretroviral therapies within the first few weeks, not too early, probably, but not too late. You know, that sweet spot has to be defined. But if you go on antiretroviral therapy at a certain point in time, you stay on it for quite a period of time, years, and you stop, maybe 1 in 10 people will not have a rebound. The virus will be suppressed for some time. These are the so-called post-treatment controllers. We don't know the mechanism. We don't know who actually will actually control virus when they stop therapy. But what we do know is that it is now possible therapeutically with antiretroviral drugs to manipulate the relationship between the virus and the host, leading to post-treatment control. And so there's a lot of excitement to try to figure out the mechanism and the predictors so that we can begin to do this in the clinic. How about shock and kill? Probably the first area of HIV cure research going back now 10 years in the clinic has been this concept that we bring people in. We recognize that the virus persists largely in these long-lived reservoirs. These are primarily memory CD4 positive T cells. They are maintained largely by clonal proliferation. They persist indefinitely. They never go away. The virus resides indefinitely in these cells. The provirus is deeply integrated within the host genome and is often quite silent, but occasionally comes out, virus is produced, and the virus takes off. So shock and kill is basically just this concept that we're going to actually therapeutically manipulate that provirus, force it out of its hiding place, and wait for the immune system to kill that virus-producing cell. And others, there have been like 10, 15, 20 studies with HDAC inhibitors, disulfiram, TLR agonist, cytokines, basically have shown pretty convincingly that we can in fact reverse latency. We can get these cells to make RNA, and we can even make these cells make protein, but the cells do not die. And there's no really effective shock and kill mechanism. But I'm optimistic, and I'm optimistic based on 2 studies which are emerging. One was recently published, this one by a group at the University of North Carolina and at Emory University. They identified what is widely perceived now as the most exciting and promising way to reverse latency. These are the so-called SMAC mimetics. They activate cells through a specific pathway, NF-kappa B, in a very unique manner. And in mice and in monkeys, as shown here, whenever you give, when you take these animals and they're on antiretroviral drugs, and you give them a SMAC mimetic, you get this robust, reproducible, immediate production of virus at quite high levels. And this is by far and away the most impressive latency reversal that happens in animals. Notably, in these studies, by just reversing latency on its own and causing all this virus to come out, there's been no evidence of any real benefit because the cells that are apparently making the virus are not dying. So we have the shock part down, at least in animals, but maybe not the kill. But in people, there is this pilot data, which for me is the most exciting option out there in the shock and kill world. And this is using interleukin-15. It's a cytokine. It turns on CD4 T cells. It also, which can activate the reservoir. It also enhances CD8 T cells and perhaps kill. And in a small pilot study done by Tim Shacker and colleagues in Minnesota, they've shown that with interleukin-15 by itself in people, you can cause latency reversal. And over time, the size of the inducible reservoir shown here can go down. This needs to be confirmed. This approach by itself is not going to be curative, but it actually provides proof of concept in people that you can reverse latency, you can do it safely, and you can in fact reduce the reservoir, perhaps through immune clearance. On the other end, there's the so-called latency silencing, block and lock. This is if you can't force the virus out, can you push it back down? We think this is biologically plausible because in the natural history of HIV infection, we and others have identified a group of people who are now termed as exceptional or extraordinary leak controllers. Those are people who have very, very low reservoirs. One person in particular that lives here in San Francisco, Maureen, shown here, she's been out, she's been public about this. She is remarkable in that, well, she's remarkable in many ways. But she's also remarkable in terms of the fact that despite having been infected in all therapy for a couple decades now, we and others cannot find any virus anywhere in her body. Shu Yu at Ragon Institute has done amazing work in working this up and has found basically that in these individuals, there's either no intact virus or the virus that is there has somehow been forced into deep latency, can't come out, often because its location is in these parts of the genome in which the virus is not transcribed. So this is a phenomenon. It happens in people. On the other, suggesting that occurs possible by somehow silencing the reservoir. In my experience, in some of the studies that we're doing in people who've been on long-term antiretroviral therapy, we're talking about 20 years. Much of the reservoir during that long period of time becomes deeper and deeper, harder to reverse. So the big question on the table, therapeutically, can we sort of do this in people? There have been some exciting data generated in mice that you can by manipulating viral proteins like TAT. But as of now, there's not a lot of activity in terms of clinical trials. I think this is one of the understudied areas. It's my hope that we'll begin to find therapeutic interventions that can achieve this outcome. Gene therapy, massive amount of interest right now in gene therapy. I could spend hours talking about it. There's a lot of investment in this arena. It's being driven by excitement in other areas of medicine. Broadly speaking, there are 4 different approaches being taken. One is genetically engineering stem cells and CD4 T cells to make them protected by HIV. Proof of concept that we can do this is the Berlin and London and Dusseldorf cases in which CCR5 basically was knocked out by doing a bone marrow transplant by a donor who naturally lacks CCR5. This is gene therapy per se, but it does provide the proof of concept that if you can knock out CCR5, and we can do this now genetically, you can rebuild an immune system that will be protected by HIV. There are stem cell studies going on, there are T cell study going on to try to recapitulate this therapeutically, typically ex vivo, taking cells from people, knocking out CCR5, putting it back in. There's the kill approach, and this is basically enhancing by inserting genes in the CD8 T cells to make them more likely to track down HIV and kill cells. We think this is biologically plausible because being done now routinely in cancer, where CAR T cells can cure certain cancers. CAR T cells are expensive, they're toxic, but there's a massive investment in this arena. The cells are becoming more effective, they're becoming easier to use, and they're becoming safer. And so CAR T cell studies are being done in HIV. Control, I'll talk about in a sec. And purge is this concept of using enzymes that you can deliver various vectors such that they get deposited in CD4 T cells around the body, and they basically disrupt the provirus that exists in the latent reservoir. There's proof of concept that you can do this in mice. The fundamental problem here, though, is if you're going to try to disrupt each provirus, you're going to have to be 100% effective, which is going to be a challenge because if you leave one provirus behind and it's replication competent, that's all the virus will need. So these are a lot of interest in gene therapy in general. At the CROI meeting in the past, a few months ago, the team at the Vaccine Research Center, building on previous work, which shown that in monkeys anyway, that if you take a vector, AAV in particular, and you inject this into the muscle of the monkeys, and you deliver within the tissues these genes for these broadly neutralizing antibodies, which have powerful antiviral effects, and these genes get taken up by these cells, and these cells make the protein, the antibodies, and they release it. In theory, you can develop these protein factories and make neutralizing antibodies that persist indefinitely, that basically act like antiretroviral drugs. You stop your meds, the antibodies take over, and basically the virus is trapped. We haven't achieved this in humans, but the Vaccine Research Center team showed in a late breaker at CROI that it is at least conceptually possible and safe to give a people with HIV this vector AAV and gene-modified tissue cells such that a powerful antibody, VRC07, can be made in a way in which it works and is a sustained approach. So there's huge amount of excitement in these approaches in general. In theory, if this works, all you would need to do is bring people in, give them a shot in the arm, and wait for these cells to start making protein, and basically people don't need treatment for life. That's an aspirational goal. Hopefully we'll achieve it someday. Finally, the biggest area, the most complex area, the most robust area right now of research is being inspired largely by what's happening in cancer immunotherapy and by major successes in monkeys. This is the concept of using combination approaches of vaccines, broadly neutralizing antibodies, vaccine adjuvants, cytokines like IL-15, immune checkpoint blockers like those that block PD-1, but using typically a combination of these various approaches with the goal of making the immune system, particularly CD8 T cells, far more effective, far more likely to be able to control HIV, basically achieving so-called elite control or post-treatment control in people by modifying and manipulating, enhancing the immune system that you can generate these T cells that are effective and persistent definitely. We're optimistic about this because of the progress that's been made in cancer, but also monkeys. And here's another late breaker from CROI. Dan Baruch's team put together a study in which they took monkeys, put them on antiretroviral therapies, gave them a therapeutic vaccine, Ad26 with an MVA boost. The goal there is to boost up the CD8 T cell responses on ART. This was followed by a combination of a TLR7 agonist, which enhanced that vaccine responsiveness, but also caused probably low-level latency reversal in the context of neutralizing antibodies, which helped reduce the reservoir by killing these cells. But apparently in these monkeys, both doing some shock activities, causing the virus to come out, and a fair amount of killing to get the reservoir down while building up a very powerful immune system. That if you did all three of these things in a complex but doable approach, as shown in the in these plots here, in the right bottom quadrant there, that if you basically do all this, vaccinate, antibodies, vaccine adjuvant, and stop therapy, a few animals don't rebound, and those animals that do rebound do so and return basically to very low levels to become controllers. We're very excited by this because we're working on with this entire approach. We're working with vaccines, we're working with antibodies, and we're working with these TLR agonists in people. For the most part, these are studies that are being done in uncontrolled manner. As shown here, for example, this is a study that was presented by our group in collaboration with others in Gilead, that was presented at CROI by Devi Singh Gupta from Gilead. She presented these data a few months ago. And what we did here is we took people who had previously been on long-term, previously controlled virus on their own, but had detectable virus. So they were low-level viremic controllers. They went on therapy, and then they came to us. And we said, okay, you once had an immune system that controlled the virus. So you have CD8 T cells that work. Can we boost them to make you go from being a low-level viremic controller to an elite controller? That was the idea. So we took 25 people. They were randomized to receive a placebo or this TLR7 agonist given every 2 weeks for 10 doses. And we stopped therapy. And there was a modest effect in terms of the rebound kinetics. People who got the TLR7 agonist rebounded about a week later. Nothing dramatic. But interestingly, during the period of time that we gave treatment, we observed that the amount of the reservoir that's intact, as estimated with these new assays that developed by Bob O'Connell and others, IPTA, we found that you can actually reduce the reservoir size during treatment. No one's ever done this in people, so we're quite excited by this. We think it's probably being mediated through innate immune responses. More about this will be discussed at the IS meeting coming up. But this provides proof of concept that a vaccine adjuvant like TLR7 can have an effect on its own. We're working with vaccines, we're working with these antibodies, others are doing that. And so in the clinic right now are a number of these sort of combination approaches, proof of concept studies, people going for the home run, people taking often vaccines and their adjuvants and so forth, mixing them up to try to build up a CD8 T cell response that can actually ultimately lead to control of the virus. Now, these are complicated studies, they're intensive, there's multiple modalities. At the end of the day, no one's gonna wanna do this if they can take a pill a day. But the idea here is that if you can actually prove proof of concept that this can work, then these things can be optimized in the future. So just in conclusion, I know that I went past it, it was superficial. We have meetings now that go 4 days that go into all this stuff that I just discussed. But I just wanted to provide clinicians in particular with sort of a broad sense of the greatest hits now in HIV Cure, where we're at in the clinic. Progress continues to be made, not necessarily so much in product development, the stuff I've been talking about, but in terms of coming up with the strategies, the preclinical animal models, and really understanding to the nth degree how the reservoir is maintained, which I did not have time to go into. As I said, multiple approaches are being tested. All have the ability ultimately to work. Right now, as being done in the clinic, none are going to be particularly impactful because they're complicated and so forth. But the hope is if we can prove proof of concept, they'll be optimized. And one thing that is just remarkable about the cure agenda is the synergies between what's happening in HIV cure and COVID research, what's happening in HIV cure and cancer research, what's happening between HIV cure and prevention of HIV or treatment of hepatitis B. Massive synergies are now actually being recognized between our field and other fields, which actually makes this a very exciting place to be working. And so with that, I'll pass it back to our moderators, and I appreciate your interest and attention. Thank you.
Thank you very much, Steve. That was a wonderful talk. Lots of stimulating questions. We'll maybe take 1 or 2 questions now, and then in the roundtable, touch on some of them. So Steve, you mentioned the extraordinary controllers or deep latency. The woman from San Francisco. Could you tell us a little bit about some of the others and how that concept might be relevant to patients who've indeed been undetectable for so long?
Well, actually, no, the last part of your question there, Jonathan, is key. So if you take people who've been on ART for like 2 decades, right? And we now have people out to 25 years. The data that's emerging suggests that the virus that's left is really deep in the latency. And the mechanisms for that are complicated and there's multiple different pathways. But it does suggest that not all latent viruses are equal. And if we can therapeutically push things a bit deeper, then we probably don't care about it anymore. Mm-hmm. And there are, I didn't have time to go out in the presentation, but there are drugs. We're particularly interested in a drug class called the Mtor Inhibitors, for which there is some preliminary data that we might be seeing that in the clinic.
And maybe just one more question on the shock and kill, which of course was very popular a while back. Sort of where did that stand? But the issue also, I remember there was a lot of discussion about sanctuary size, the CNS, what would happen to CNS. If this moving forward, are those concerns, have they been addressed at all?
Well, there was not really. To be honest, we don't really have any idea what the safety of this approach will be because we haven't really achieved it yet in people. I mean, we have, to be honest, we really have not really achieved the shock effect. We've got tiny signals that I discussed that suggest that we're going to get there. All I can say is that in monkeys, when you do successfully shock that virus out and get a fair amount of virus, we're not seeing any evidence of any clinical effects. And I personally think the amount of virus that's produced in these types of settings is never going to be dramatic. It's, the viral loads that we see at most are going to be a couple hundred copies. And we know from clinical experience that people sort of tolerate that. So that's not one of my concerns moving forward.
Great. Well, Steve, we'll move to the next topic. We're getting a lot of questions and we'll try to address them in the roundtable. Thanks a lot.
Thank you.
So we'll move to our next speaker. Linda-Gail Bekker is former president of the International AIDS Society. She's deputy director, Desmond Tutu HIV Center, Institute of Infectious Disease and Molecular Medicine, University of Cape Town, and is the chief operating officer of the Desmond Tutu HIV Foundation. Dr. Bekker is principal investigator of UCT's Clinical Trials Unit and has chaired protocols for the HIV Vaccine Trials Network and the HIV Prevention Trials Network. Additionally, Linda heads the Desmond Tutu Center of Adolescent Health and Well-Being. Without doubt, Dr. Bekker's been at the forefront of the global fight against HIV, and today is one of the leading researchers in HIV vaccine development. And we're delighted to have her speaking with us today on HIV vaccines. Linda?
Hello, everyone. I'm very pleased to have this opportunity to bring you up to date on HIV vaccines. Where are we? Indeed, the world does need an HIV vaccine. And what could we imagine the perfect profile of that vaccine might look like? Well, we think it should ideally have come in a single dose, be durable enough to provide a lifetime of cover, have minimal side effects, cross-clade protection, be administered simply, and really have very few issues on the logistics and handling side. Ideally, we would co-administer it with other vaccines. But in fact, modelers have looked and shown that even a partially effective vaccine, if rolled out sufficiently across the world, would make a significant difference in the epidemic as we know it today. But in fact, it has taken a very long time for us to have an HIV vaccine. And as you know, we don't yet have one. Why has it been so challenging? Well, we know that this pathogen, HIV, has more genetic diversity than any other. We don't have any natural immunity to HIV infection, and there's no human challenge model. Our animal models are imperfect, and there is unfortunately a perceived limited return to investment to develop vaccines. And so consequently, vaccine development has been particularly slow with HIV, although it must be said, for many other vaccines, this has similarly been a problem. We can take two different approaches to vaccine design. One is an empirical or inductive approach, where we test promising candidates until vaccine efficacy achieved. We then identify correlates and optimize the regimen. The other is a theoretical or deductive approach, working in the opposite direction, where we optimize the immunogen and the regimen, and then work back to figure out what the vaccine should look like in order to cause that particular vaccine response. Of course, there are a number of vaccine approaches that are at work today. Our history over 30 years has been really one of waxing and waning in terms of hope and optimism when it comes to HIV vaccine development. The early trials involved the VaxGen products, one done in the US, one done in Thailand, both of which showed no evidence of protection, despite really showing good participation of communities to get involved and no safety issues. The second era of vaccines was based on a T-cell approach, the hypothesis being that CD8 T-cells differentiate elite controllers from progressive HIV infection. And therefore, if vaccines could induce such responses, they may be effective in reducing HIV acquisition or post-acquisition viral load. So the hypothesis was the more potent the T-cell response, the better the vaccine. And that again involved a couple of trials, the STEP trial, and notably carried out in the US, HVTN 502, and its sister study, PharmD, in South Africa. Both trials were large, and the approach was to stimulate T-cell responses, but in both cases, the trial was stopped by the DSMB after a preliminary analysis that showed that the vaccine would not meet its endpoints. More concerning, in some participants, the vaccine may have made them more vulnerable to infection. HVTN 505 came out of those studies, also using the Ad5 vector platform. But in this case, eligibility in the participants was a low adenovirus seropositivity, and men had to be circumcised. Again, unfortunately, stopped by the DSMB for futility. Then, towards the end of the first decade of 2000, we had the first light at the end of the vaccine tunnel, the RV144 trial, a regimen of LVAC priming followed by GP120 boost. Extraordinary study carried out in Thailand among 16,000 individuals. And the results of that study were met somewhat with skepticism and a great deal of surprise, because they had used two vaccine candidates that before had shown no promise at all. But in this trial, we saw a 60% reduction of HIV acquisition after 12 months in the modeling study, and at the primary endpoint of the study at 42 months, a 31% reduction in HIV acquisition. This led to a flurry of efforts to find the immune correlates that led to this efficacy. In clinical trials that show the efficacy of a vaccine, the identification of immune responses that are predictive of trial outcomes generate hypotheses about which those responses are responsible for protection. And to determine whether a correlate of infection is a cause of vaccine protection, it must again be tested within an efficacy trial. And so those immune correlates were found in an extraordinary worldwide effort, and were found to be reliant on antibodies to the conserved region of V1, V2, that there was no direct correlation between neutralizing antibodies and HIV acquisition, and the polyfunctionality scores of the envelope-specific CD4 T cells were also key to the correlates. So taking those lessons forward gave rise to a number of new vaccine trials. And I'm going to go through them very briefly in the next few minutes. First of all, there was the P5 grouping who built on the RV144 trial, leading to the HVTN 702 study. And I'm going to describe that first in more detail. So building on the RV144 trial, taking the LVAC and GP120 forward, the candidates were modified for a South African scenario. And this meant changing the immunogens to be more in keeping with the clade C epidemic. In addition to increased potency, the adjuvant was changed from what was used in the Thai study to MF59, and an extra boost was added. So the strategy for the P5 program, which was really a public-private partnership to bring the RV144 trial results forward, resulted in 3 trials, the HVTN 097, which really looked at the same candidates in a South African scenario, HVTN 100 to test the new clade C changed candidates in South Africa, and then ultimately leading to HVTN 702. I'm not going to discuss 097 here, but suffice to say that we saw the same, if not better, immune responses when we brought the Thai vaccine candidates to South Africa. So this is HVTN 100 schema, and it was the forerunner to the efficacy trial. So as you can see, the same products used in RV144, now with clade C inserts, with an additional boost at month 12, and an additional boost thereafter, again at month 18. And 100 went into the field first to explore the go, no-go criteria for an efficacy trial. And it was required that 100 meet all of these various criteria in order to allow the efficacy trial to move forward. And this has been published now, and all those go, no-go criteria were met. As a result, we moved on from 100 into 702 with the lessons that it looked like the candidates could elicit the required correlates, that we would require additional boosting for durability and for potency. So 702 was conducted only in South Africa using these candidates. And what you see here is 5,400 South Africans were enrolled in the trial with that schema as suggested. The trial sites were in South Africa, and 14 sites were activated with a preponderance of young women in the sites. The study accrued very well with very good retention, was very well conducted, but unfortunately in early this year, the DSMB met, and this unfortunately triggered a teleconference that led to the belief that the trial would not reach the efficacy endpoints, therefore futility, and so the recommendation was that vaccinations should be paused. This is the important result. Over 36 months, you see no difference between the placebo and the vaccine arms. Importantly, there was no evidence of harm, which is something we always worry about in vaccine trials, but also very unnerving. When we looked at the difference by sex, you just see this extraordinary incidence amongst young women in this part of the world, in South Africa, really, I suppose, underlining the need for better prevention to stop HIV. Moving quickly to the second efficacy trial, this is involving the Johnson & Johnson Ad26 product. This is an interesting vaccine regimen which seeks to aim for global coverage. So it does involve the Ad26 mosaic and is multiclade and designed to have an impact around the world. So polyvalent mosaic proteins are assembled from natural sequences by in silico recombination and optimized to provide maximal coverage. So the phase 2 study showed a good efficacy and good response. And so quickly, the phase 3 studies were brought about. The first HVTN705, known as Imbokodo, is again in the southern African region and involves young women who have sex with men. This is the schema. There are 2,600 women involved in this placebo-controlled trial. And the trial sites are all around South Africa and Eastern and Central Africa, as you see. The trial was fully accrued in mid-2019. 2,637 participants in 23 sites. And of course, in early March, as everyone knows, we were hit with the first COVID cases in South Africa. This led to a scramble to make sure that we would get everybody vaccinated before any lockdowns occurred. And I'm pleased to announce that by the 1st of June 2020, just about every single participant had their full regimen of vaccination. And so this study is in its final stages of follow-up now, and hopefully will show good results. The Mosaico is its companion study being run in the Americas and a number of other countries in the north. 3,800 participants targeting particularly men who have sex with men and transgender women. And this here you see the global site distribution. This study was really just kicking off as COVID hit, and so has been on pause, but will slowly begin to reopen as the world comes back to some kind of normality, and we look forward to seeing Mosaico unfold. The final set of studies involves antibodies, and there has been, this is the passive transfer of antibodies, so passive immunization. There is a long history of using antibodies to treat and prevent infectious diseases. And of course, it isn't new, but this is the first time we are exploring it as a prevention technology for HIV. So as you know, identifying broadly neutralizing antibodies can either be used to design immunogens or can actually be used as therapy or prophylaxis to stop a particular infection. And the last 10 years has given rise to an extraordinary set of discoveries of very powerful neutralizing antibodies. The one that has first been selected for further investigation is known as VRC01, which targets the CD4 binding site on GP120. And VRC01 has been shown to be very effective and to have significant potency. For this reason, it has been brought forward into a clinical trial known as the Antibody-Mediated Prevention Trial. There are 2 cohorts, one of men who have sex with men and transgender women in North and South America, and the other one amongst women who have sex with men in sub-Saharan Africa. The schema for these trials is it's placebo-controlled. These are intravenous infusions carried out every 2 months over a period of time. And here you see there are 2 doses that are being investigated. This is a proof of concept trial. It is fully accrued. It is in follow-up, and the results are expected later this year. So we are very excited to see if this is potentially a way forward for HIV vaccinology. As mentioned, there are a lot of exciting antibodies following close on the heels of VRC01. And here you see where they act on the important components of the HIV virus. Excitingly, some of these have been combined into tri-specific antibodies. And again, these are now going into clinical trials for the first time, and we'll be seeing the impact not only of one antibody, but of a mixture of broadly neutralizing antibodies to prevent HIV. And so these antibodies are at various stages of testing in humans. I mentioned that there is another way that we can design immunogens, and that is to work backwards, to engineer vaccine design by coaxing the right broadly neutralizing antibodies. And the first of these trials are now coming to the end of their phase 1, showing safety and proof of concept that this actually can happen and can be done. So I'm saying to all of you, watch this space. It's an extraordinarily exciting aspect of vaccine discovery in HIV, and I think we'll be hearing much more. So in summary, we have a relatively full vaccine pipeline. We have non-neutralizing immune responses. We have broad T-cell responses also coming into the fore. We are using viral vectors and neutralizing immune responses or BNAB. So all of these moving along in our pipeline. And I would say it's an exciting time to be in HIV vaccine discovery. The science is advancing through clinical trials. We have had a disappointment in the last year in terms of 702, but there are other trials in the works and moving along. There are scientific advances fueling vaccine discovery, and I've hinted at some of those. And of course, we have the next generation of vaccines entering the clinics. I think we still want to ask, do BNABs protect? What is the potency and durability of our vaccines, and how much is variability going to matter? And can we get these BNABs to maturate in the way that we want them to? It has been a long road, more than 30 years. And of course, prevention has come on, as has treatment. We're now talking about cure and remission and very exciting prevention options. And it's likely that the future is one of an integrated prevention response, more than one modality being used in combination or for individuals to choose from. And I think it would be wrong if I did not record that other wonderful new innovations have recently been found that I think make it an important time when we need to collaborate, we need to find efficiencies, creativity, and keep engaged with community. This study, PrEP-Vac, also in the field, looks at the possibility of combining both oral pre-exposure prophylaxis and vaccines. And so I think PrEP-Vac really leads the way in terms of us understanding how to combine prevention. We are in the midst of a prevention revolution. We need to have better primary prevention, and I think it is an exciting time to be thinking about this. Suffice to say, many of us are also turning our minds to finding a vaccine for SARS coronavirus, and I'm very excited to see that vaccinology can be sped up. We are watching Operation Warp Speed in process, and we're told that we may have an effective vaccine for COVID as early as the beginning of 2021. We hope some of the lessons that we learn from HIV are being applied in this field, and we hope that those same lessons will be reapplied to HIV once we get beyond this pandemic. Really want to acknowledge a lot of good people who have helped me put this talk together. And I note the Global HIV Vaccine Enterprise that works hard to find an HIV vaccine for the world. Thank you very much.
Thank you, Linda. That was a wonderful overview and gave us some optimism. So that was a real treat. try to take one or two questions, and then if we have time, the roundtable. So I think something that has come up when people are speaking about COVID is the duration that the vaccine will last. Obviously, people are sexually active for many, many decades. And if we do have a vaccine, will this be lifelong? Will it need to be revaccinated every few years? Is there any indication on differences? Has that been considered?
Hi, Jonathan. You know, I think that is our concern, particularly when we've just had the wonderful breakthrough that I was alluding to of the CAB long-acting, where people are going to be able to come in every 2 months for another shot. You know, where does pre-exposure prophylaxis and an HIV vaccine begin to overlap? And I think the hope is that even if our prototype, if our first vaccination that is effective does require quite a few boosts, down the pike, we might be able to hone that. I think ultimately we are looking for something perhaps more like the anti-tetanus approach, where every 10 years, maybe somebody needs a boost, or, you know, we vaccinate a pre-adolescent, get her through the difficult adolescent era, and maybe she only requires another boost in her mid-20s or something along those lines. So, you know, that ultimately is what I think a vaccine is going to have to do if we are going to compete with the long-acting antiretroviral. So we keep holding on to that hope.
So maybe one last question. Folks voice concerned about the clades. You know, that some of the vaccines do keep that in consideration, some don't. How relevant is that really for the difference? Some clades are very different. How different are they actually at the targets we're looking at? What do you think on that issue?
Well, you know, the world sort of divides into two groups in this regard. I thought it was really exciting in that 097, and with all of these talks, one has to go over this data so quickly. But in HVT in 097, we took the clade BE that had been used in the Thai trial, we brought it to South Africa, and we put it into our clade CE population here, and actually saw better immune responses than what they'd seen in Thailand. So kind of suggesting that clade is going to be less important. The nice thing about the AD26 mosaic is that it's multi-clade, multi-gene, and so, we're not going to care there as much, I don't think. But yes, I think the clade issue sits in the back of our mind, and it does mean we want to test these vaccines in multiple different settings to be sure that we're going to see the same kind of results should we see result in one part of the world compared to somewhere else.
Wonderful, thank you. So I think we'll get back to the issue you mentioned of how prevention will PrEP and vaccines, and we'll touch on that, and we'll move to the next topic and wait for the roundtable. Thank you, Linda.
Thanks, Jonathan.
So our next speaker is Dr. Richard Elion, who works in the Department of Health in Washington, D.C. as co-director of their HIV prevention efforts in the Sexual Health Program, and with the Washington Health Institute of Director of Clinical Research. Rick is currently an associate professor of medicine at George Washington University and at Johns Hopkins University. Rick has been active in HIV care and prevention from very early in the pandemic and has penned some of the important research both on treatment and in recent years in prevention, both doing research, but very, very much still involved at the front lines. He's one of the world's experts, and we're delighted to have him speaking to us on oral versus injectable PrEP. Rick?
Hi, I'm glad to be with you today because I want to discuss HIV prevention, some of the new emerging options. There are currently new choices developing for HIV prevention that are very exciting. In 2012, we started to see the development of oral agents, TDF-FTC. 2019, that progressed to 2018, 2019, TAF and FTC. And now we've currently got emerging injectable cabotegravir that might be coming. So the issues about oral, long-acting oral, injectables, implants are all emerging choices that I want to discuss with you today. We know that currently the population that is appropriate for PrEP is any of these groups that has behaviors that put themselves at risk. And so this helps establish the population of people who are relevant for HIV acquisition. And we know that in various projects across the world, we've seen the benefits of HIV prevention, from High's reduction of close to 60% in San Francisco to at least a third reduction seen in Sydney. And so there's no question that the impact of PrEP on the development of HIV has been a significant factor. The question then becomes, what have been the existing agents? We know that the currently approved FDA agents in the States, and I believe in Europe as well, are tenofovir FTC, and for those with creatinine clearance greater than 60, and tenofovir alafenamide FTC, and that goes down to creatinine clearance of greater than 30. Both of these, and the TAF version is not available for people people who are at risk for acquiring HIV from receptive vaginal sex, so not for women predominantly. It hasn't fully been tested. And so these establish the two oral regimens that have been used. So what are the pluses and minuses of these oral agents? When we look at TAF, we see that TAF, which was developed because it had a better renal and bone sparing profile than TDF, which was associated to have some impact on kidney and bone. You see that it was just as effective at preventing HIV acquisition as TDF-FTC, arithmetically even better, but statistically non-inferior. So it establishes both FTAF and TDF-FTC as reasonable oral options to prevent HIV acquisitions. The benefits of TAF have to do more with that sparing of bone demineralization and more gentler on the kidneys. Another way that TDF-FTC was explored to be used instead of on a daily basis is the idea on event-driven or sex-driven PrEP. And this, you would take 2 tablets 2 to 24 hours before sex, 1 tablet 24 hours, 1 tablet 48 hours. So a total of 4 pills. And this approach in subsequent analysis was shown to be statistically virtually the same as daily PrEP. So this establishes on-demand PrEP as an alternative for those who don't wish to take PrEP on a daily basis. And so I think we start to see that the oral agents can be used in different ways, either daily or on demand, and also with different populations in mind. We know that one of the key elements of taking TDF and TAF to some degree is the issue of adherence. In this study, you can see that once people start to miss more than 2 pills, 2 to 3 pills in a given week, once they're in steady state, you have a higher range of HIV incidence. And so it stresses the need that you've got to be taking close to 4 to 5 pills every 7 days, and you can't miss more than that. And when there's periods of time where more is missed, you are at risk. When we look at the impact of that risk, to look at the development of drug resistance, we see that for the most part, resistance is seen in patients who start their PrEP when in fact they were HIV positive, that they enter on the window period of the test where the test hasn't detected HIV yet, but the individual already is HIV positive. Those individuals are at risk of development of resistance. And so that's why we're very careful about the use of proper HIV testing to determine who's actually HIV negative before starting. For those people who do start, we do see, however, that the incidence of rate of resistance is very low. And looking at these seroconversions, for the most part, it's only going to be predominantly the M184V or resistance to emtricitabine that is seen, with very few patients showing resistance on tenofovir, but still a very low rate of these. And so the CDC recommendations are for anybody who has a break in treatment to assume they could be HIV positive, make sure you test them and start them over again as you would somebody just beginning PrEP. The bone sparing and kidney sparing has been discussed. This gives you a way to look at some of the improvements in people who stop daily PrEP, that you see improvements. And if you were to put patients on TAF, you see very similar to what you see in the lines in green. You see, for the most part, very well tolerated. So one of the challenges of TDF-FTC for those at risk of bone demineralization is this kind of change in bone function, and also some declines in GFR. One of the challenges about the use of TAF instead of TDF has to do with weight gain. There's been a lot discussed about this in terms of treatment, but this is in prevention, so looking at HIV negatives, and you can see over a 2-year period about a 1.7 kilo or 3-pound gain compared to about a 1-pound gain on FTAF. And so that's not a huge amount of weight difference, but it would be might be relevant for some people, but for the most part, I think this is a comforting stat to see not a big weight gain on TAF. Perhaps with oral agents, one of the concerns we have is about retention. Maybe oral agents are harder to stay on. And so we see here data from the San Francisco clinic. We're looking at the end of the 8 months, only a little less than 40% of the patients who started PrEP stayed on it. The alternative understanding of this is that perhaps people only take PrEP when they need it, and that it's not about the oral agent, but about the individual experience of times of risk, seasons of risk, relationships end, your interest wanes. And so it's possible these adherence, these engagement or declines in engagement with PrEP decline over time naturally, not based on the modality. However, various approaches have been done to help test that question, and looking at that, but through the use of navigators and other counseling and community for populations at risk. Another approach is, instead of waiting to start PrEP, to start it immediately. We do that at the Department of Health in DC. This is a study looking at this in New York City, and only 3% of the candidates had to be delayed. 97% qualified, and you don't give, you only give a short amount of drug for people who have a rapid test that's negative. They come back a week later, and then they get review their labs, and then they get their 1 month or 3 month supply as the protocol indicates. So this is a way to help improve the modality of giving it, not just the approach. The benefits of oral agents have been well described in the last 10 minutes, but I'd like to focus now on some of the newer developments. And this is the development of injectable and longer acting agents. The particular one I want to start with is cabotegravir, and you can see here in HPTN 083, this was the study design that looked at the use of giving this injection every 2 months compared to TDF-FTC. This study was recently discontinued because of significant differences in the rate of new HIV seroconversions in the population who got TDF-FTC with a higher rate of conversions compared to cabotegravir. This study looked at 4,500 cisgender MSM and TG and transgender women who have sex with men. 66% were under the age of 30. Half of them were African American or Black, enrolled in the United States. About 12% were transgender women. So this was a population at definite risk. And what we saw was that the seroconversion rate was 3-fold higher in the TDF-FTC arm compared to cabotegravir. Both regimens were basically effective. The HIV conversion rate was low. However, it was higher in the group that got oral agents, and that's why the study was discontinued. In terms of safety, both arms were safe and well tolerated. There were injection site reactions, elevated temperatures, higher blood pressure in some of the patients who got the injectable cabotegravir, but not enough to cause discontinuation of the treatment. Subsequent ongoing analysis will report the timing and incidence of resistance for all HIV seroconversions, because this is an important issue. One of the issues around long-acting agents is that they stay in the blood for a long time after you discontinue treatment. So for those on cabotegravir, they had to take oral agents, TDF-FTC, for at least 12 months after the cessation of the cabotegravir to prevent the development of resistance as the levels of cabotegravir decline. How that'll influence patient selection and patient desirability of using an injectable that still requires you to take an oral agent is an open question. So the study was closed. Patients will be offered either to go on open label and will be able to go on open label cabotegravir as soon as it's available. There are other agents besides cabotegravir. I want to report on 3 others. The first is Gilead Sciences' capsid inhibitor. This is an agent that prevents the release of subsequent virions through interference of viral replication and the release of the capsid, which is the shell of the virus that goes out into the system. And what you can see here is that this drug over time had significant viral suppression for significant amounts of time. And so this agent allows us to view a prolonged dosing schedule that might be very effective for the prevention of HIV acquisition. And those studies are being developed now, and it might be that you'll take an oral agent, it might be a subcutaneous, or it might be within an implantable reservoir. Time will tell. The next agent I want to talk about is a nucleoside reverse transcriptase translocation inhibitor. So this is a new mechanism, sort of an NRTI, but a more modern version, developed by Merck. It has an active half-life of 80 to 130 hours, and a single dose suppressed HIV for greater than 7 days, and you can see this dose response curve. In PrEP settings, it's possible that this could be given orally or in an implant with extended use applications. This study is looking at an implant of islatravir, and you can see that this gave effective levels to have HIV prevention upwards of 12 weeks. And so this starts to give us the idea of a not just 2 months, but perhaps 3 months, where we could see prevention of HIV acquisition. Islatravir is very well, for the most part, has appeared well tolerated and was effective. And so it really gives us a lot of hope for a new class of agent. And it may be that we would dose prime somebody, and then it would allow for a different dosing schedule. The exact dosing is still to be worked out in upcoming studies that are starting this year. And finally, I want to close with the idea of using broadly neutralizing antibodies. These have first been looked at as a treatment intervention to help decrease the amount of time people need to stay on ARVs by using these antibodies either in lieu of ARVs or in addition to ARVs. However, they also have applications in terms of prevention, and that by giving these intermittently by injections or IV, maybe perhaps 2 months, every 3 months, and I think the interval between treatments is still being evolved, these can also be an effective way to prevent HIV acquisition. And so finally, we get to how we're going to make choices. The issue is, how do we match individual patient preferences with the scientific profile of these different issues? What issues of weight gain will be relevant for current oral agents? Will that be a deal breaker for some people? Will renal and bone be so relevant that it'll dictate which regimens people should do of oral agents? What will be the aspect about resistance as we look at the difference between oral and injectables? Will the necessity to have a tail for an oral agent for a previous injectable approach be a deal breaker for some people, or not that big a deal of a transition? Will bi-monthly injections be better for individuals than a daily pill? We know in the world of oral contraceptives, we've seen a whole variety of consumer responses. For some people, the answer is absolutely. The difference in time intervals is critical. For other people, taking a daily might be better. But what if we have an implant that could be done every 6 months or every 3 months? Is there a critical time period of interval between treatments? These are the emerging study questions that we hope we'll get answers on in the next 12 to 24 months. But the bottom line is that since 2012, when we had the first advent of HIV prevention strategies, we've come a long way to 2020. And the next 2 to 3 years will give us even more choices. And so our role as clinicians will be to match these opportunities of patient preferences with the scientific advantages of the emerging technologies. I'm happy to take questions at this time.
Great, Rick. Thank you very much. That was a fantastic talk, and actually very optimistic about the future of prevention. Looks like a lot of drugs and modalities in the pipeline. For the near future, where we may be having long-acting cabotegravir, and you mentioned some of the trade-offs, but the practical level, sort of, who do you see as the patient who would be more appropriate to stay on the oral PrEP, and what might be the characteristics of a patient you would probably consider actually going to the long-acting?
Well, that's the million-dollar question, Jonathan, because I think that the first appeal is that less frequent dosing is for less adherent patients. Mm-hmm. However, the corollary of less frequent dosing, if you don't return, leads to more resistance. So I think we're going to really have to understand that issue about how resistance develops in long-acting injectables before we can really customize it for an individual adherence profile.
Do you think it'll basically be folks who are already sort of on PrEP and doing well, and then they'll be rolled over, or will this be something that clinicians will actually take patients they didn't think were eligible and then start this as a sort of new modality?
I think it'll be a hybrid. I think that the earliest adopters will be those people who are already adherent, who wish to take a regimen that they have to do less frequently. On the other hand, I think as Linda's presentation pointed out, we're starting to get a confluence of the issues between what is a vaccine and what are injectable preventive approaches. And so I think there's going to be populations who we can only access every couple months that are going to be appropriate for this. So I think it'll be different groups of populations, a heterogeneous group adopting these two modalities.
Yeah. And just a final, because I think we'll have a nice discussion on that issue of overlaps and the synergies between vaccines and PrEP. But the one issue which seems to come up also in the previous session, that 12-month that you'll need to take oral, I mean, will that be a large, I see that as a barrier to many patients actually taking the leap of taking for prevention cabotegravir.
Well, it will be if it doesn't change. And there's a lot of controversy about how long that extended tail has to be. And we get into the sort of human experience. If we can cut that to 6 months, does that make it more tolerable? And I'm not sure that 12 months would be the earliest answer. I think that's been the design in the studies because of PK studies that we've seen, but I have a feeling that'll get shorter over time.
Sure. Well, thanks, Roderick. We'll look forward to the roundtable after the next talk. Thanks a lot.
Thank you very much.
So with that, we'll move on to our final speaker. We have a case presentation. I'm delighted to introduce Catia Marzolini, Associate Professor in the Division of Infectious Disease and Hospital Epidemiology at the University Hospital of Basel. Dr. Marzolini is also a member of the Liverpool HIV drug-drug interactions website and is notably in charge of predicting drug-drug interactions. Catia is a member of the European AIDS Clinical Society Guidelines panel for the pharmacological aspects of HIV therapy. Her research focuses on pharmacological and genetic determinants of drug response and toxicity. I can tell you personally that Catia is a wonderful resource for many of us clinicians faced with challenging clinical pharmacological dilemmas, and she's also a very gifted teacher. So there's no one better suited to finish our session by presenting a case on potential challenges of drug interactions for long-acting agents. Catia?
So good evening. I would like to thank the organizer for the opportunity to present this case, which illustrates drug-drug interaction and other pharmacological consideration for the long-acting cabotegravir used as PrEP. So I will start first with the case presentation. This is Adrian, a 29-year-old MSM. Adrian is currently on oral PrEP with TDF and FTC. He has no renal impairment, is overweight. He does do chemsex, takes occasionally antacid and St. John wort for a mild depression. So Adrian came across a recent press release mentioning that oral long-acting cabotegravir was more efficient than oral PrEP with TDF and FTC to prevent HIV infection. And this statement relates to the study HPTN-083, which aims at evaluating the efficacy of long-acting cabotegravir in MSM and transgender at high risk of HIV acquisition. And actually, the Data Monitoring Board recommended to stop earlier the blinded part of the study because the results show a 3 times lower risk of acquiring HIV infection in the cabotegravir long-acting arm as compared to the oral PrEP, likely due to a lower adherence to the oral PrEP. So Adrian is very much interested in the concept of receiving an injection every 8 weeks and was wondering whether he would qualify for such a prevention treatment in the future when it will be available. So before discussing drug-drug interaction, Adrian, who is overweight, has expressed some concern about gaining weight with cabotegravir, while he heard that some HIV integrase inhibitors can lead to a weight gain. So what do we know about cabotegravir and weight gain? Actually, the study HPTN-077, which aimed at evaluating the pharmacokinetic and tolerability of long-acting cabotegravir as PrEP, were analyzed and showed that there is no significant difference in terms of weight gain between the cabotegravir and placebo arm. Also, there are no differences among ethnic group in terms of weight gain or among gender. So it looks like weight gain might not be an issue for cabotegravir. This weight question brings other important considerations. So first of all, cabotegravir injection is performed in the gluteal muscle. And it is important to know that the muscle has a rich vascular supply, which favor drug absorption. On the other hand, subcutaneous adipose tissue has a less vascular supply, which may result in less drug being absorbed. So the injection technique is absolutely critical to ensure that the drug is deposited in the muscle. And if you look at the Canadian label for Cabenuva, you can see that it recommends to use longer needle lengths in order to make sure that the drug is distributed in the muscle, because obviously patients with a higher body mass index have a larger adipose layer. Obesity can also impact the pharmacokinetic of cabotegravir, and this has been actually demonstrated in the FLAIR and ATLAS study. And these studies are aimed to evaluate the efficacy of long-acting cabotegravir with long-acting rilpivirine for the treatment of HIV infection. As a reminder, the patient underwent first an oral lead-in phase, followed by an initial intramuscular dose where cabotegravir was administered at 600 milligram and rilpivirine at 900 milligram at week 4. And then they had a maintenance intramuscular dose where cabotegravir was administered at 400 milligram and rilpivirine at 600 milligram at 8 weeks and every 4 weeks thereafter. And you can see here the trough level of cabotegravir and rilpivirine over time. At week 4, this is the trough level after the last oral dose, and at week 8, the trough level after the first intramuscular dose. Now, interestingly, when you categorize the trough level based on body mass index below or above 30, you can see, interestingly, that the median cabotegravir level were 46% lower in obese 4 weeks following the first injection as compared to non-obese. And this difference is likely due to a lower cabotegravir absorption, resulting in initial lower trough that increase nevertheless over time, and there is no difference in the trough level between obese and non-obese at week 32. Interestingly, rilpivirine does not seem to be affected by body mass index. Now, if you categorize the trough level based on gender, you see again that there is a median cabotegravir level that is 40% lower in female compared to male 4 weeks following the first injection, whereas rilpivirine, as before, does not seem to be affected by gender. Now, I'd like to point out this meta-analysis, which looked at the effect of gender on the pharmacokinetic of drugs administered orally or intravenously. And you can see a completely different picture than before, in that women tend to have slightly higher Cmax and AUC as compared to men. And this is explained by differences in body weight and metabolism, resulting in reduced first-pass effect or hepatic metabolism. And I like to make a point here, because this is actually a question that comes very often in the HIV medicine, and which is, does gender actually impact drug pharmacokinetics? So based on this data, we can say that there is no significant differences, at least clinically significant differences, in the pharmacokinetic of oral drugs, including antiretroviral drugs, in women as compared to men. So there would be no need to adjust dosage based on gender. So after this consideration on the weight and gender, I will now talk about drug-drug interaction with cabotegravir. So first, I'd like to mention that cabotegravir is mainly metabolized by UGT1A1. It does not inhibit a cytochrome or UGT enzyme. It is a substrate of PGP and BCRP. However, important to mention that inhibitors of this transporter will not cause a clinically significant interaction with cabotegravir due to its high intestinal permeability. Important also to know that cabotegravir does not inhibit lead to a significant effect, the major transporters that are involved in drug metabolism. So now looking at ecstasy and methamphetamine, these substances are metabolized by CYP2D6, but considering that cabotegravir does not inhibit or induce cytochrome, so there is no risk for cabotegravir to interact with these substances or any other chemsex drugs. What about antacid? Well, if you administer a drug orally, well, you can have drug-drug interaction that occur at the level of the intestine via, for instance, drug chelation. Cabotegravir, like any other integrase inhibitor, has the potential to chelate with divalent cations such as aluminum, calcium, that are contained in antacid. And this will actually form a complex at the level of the gastrointestinal tract so that the drug won't be absorbed as properly. But you can manage this interaction by separating the intake of cabotegravir from antacid. So just think about this interaction when you start the oral, the lead-in phase with cabotegravir, but of course this interaction will disappear as soon as you start the intramuscular injection, because in this situation you will bypass the gastrointestinal tract, and so that you won't have an interaction that can occur at the intestinal level, whereas interaction at the hepatic level can still occur. So another important characteristics about intramuscular administration of drug is the flip-flop pharmacokinetic, whereby the rate of absorption is slower than the rate of elimination. So basically, in case of intramuscular administration of drugs, the elimination half-life will be essentially governed by the absorption, whereas in the case of intravenous administration of drugs, the elimination half-life is mainly governed by the drug clearance. So in presence of a strong inducer, the intramuscular administration of drug will actually result in a reduction of the AUC because the elimination rate is increased, whereas the elimination half-life will mainly remain constant because it is controlled by the absorption rate. So this concept is actually nicely illustrated with the interaction of the strong inducer rifampicin. So you can see that rifampicin co-administered with oral cabotegravir results in a substantial decrease in cabotegravir exposure and in his elimination half-life. And this interaction was also simulated using cabotegravir long-acting. And you can see a substantial reduction in cabotegravir exposure, although to a little bit smaller extent compared to the oral administration of cabotegravir. And you can see, as mentioned before, that the elimination half-life stay basically the same. So given the substantial decrease in cabotegravir exposure with both oral and long-acting administration, it would not be recommended. It is contraindicated to use cabotegravir with strong inducer, whereas you can see that you would be able to administer oral PrEP with TDF FTC with strong inducer. So what about the co-administration with moderate inducer? So this has also been studied with oral cabotegravir being co-administered with the moderate inducer rifampicin, and you can see a modest decrease in cabotegravir level, but the concentration of cabotegravir are still well above the concentration that were shown to maintain HIV suppression in phase 2 studies. So this combination of drug would not be considered to cause a clinically significant interaction, so it's possible to co-administer cabotegravir with moderate inducer. So one of the current gaps is drug-drug interaction in special populations. So you might wonder whether the use of moderate inducer could become relevant in a obese human in the early phase of cabotegravir injection. As we have seen before, there is an initial decrease in the trough of cabotegravir after the first initial intramuscular administration. So really based on the fact that intramuscular administration of drug actually result in a lower magnitude of the DDI, I really don't think that using a moderate inducer will further decrease the cabotegravir level. As you might know, there are ongoing phase 3 studies which evaluate the efficacy and safety of cabotegravir long-acting compared to oral PrEP in women. So I think this study will inform us whether the current dosing provides sufficient drug coverage in a special population. So what about the co-administration of strong inhibitor of UGT1A1 such as atazanavir? So this drug association has also been evaluated using a PBPK model and showed that there is only a 10% increase in cabotegravir exposure, which is clearly not clinically significant. And actually, these results are also supported by another study that looked at the effect of genetic variation in UGT1A1, a genetic variation that results in a reduced function of this enzyme, and which showed that having a genetic variation in UGT1A1 will only modestly increase the oral cabotegravir or intramuscular cabotegravir exposure, which confirmed that really a strong inhibitor of UGT1A1 will not cause a clinically significant interaction. So Adrian is also considering undergoing gender reassignment and wonder whether cabotegravir could interact with feminizing hormones. So there are actually drug-drug interaction studies that were performed with hormone, essentially hormones that are used for contraception. And you can see that the oral administration of cabotegravir with levonorgestrel or ethinyl estradiol resulted in no clinically significant interaction. Of interest, the effect of various contraceptive methods was evaluated on the pharmacokinetic of long-acting cabotegravir using the data of the HPTN 077 study. And you can see that oral contraceptives were associated with a lower cabotegravir peak, but did not have any effect on the other pharmacokinetic parameters of cabotegravir, so that this interaction is considered to be likely non-clinically significant. Also, other contraceptives caused no significant effect on the pharmacokinetic parameters of long-acting cabotegravir. So based on this data and considering the fact that cabotegravir does not inhibit or induce drug metabolizing enzyme, we do not anticipate any clinically significant pharmacokinetic interaction between cabotegravir and hormones that are used for gender affirming. Now, I'd like to mention that feminizing hormone therapy has been associated with an increase in endogenous nucleotide. As you might know, endogenous nucleotide compete with tenofovir phosphorylation. So this resulted in actually in a median tenofovir diphosphate endogenous nucleotide ratio that was 7-fold lower in the rectal tissue. The ratio was not modified in PBMC. Also, the ratio of phosphorylated N-tricitabine compared to its corresponding endogenous nucleotide was not modified both in the PBMC and rectal tissue. So the clinical significance of this finding still needs to be evaluated. But I'd like to mention that there was a recent pharmacokinetic study in transgender women using oral PrEP with FTC-TDF and taking feminizing hormone, which show a modest lower reduction in the exposure of tenofovir. However, the tenofovir diphosphate level were not modified in the PBMC, and the author of the study concluded that there is no need to adjust oral PrEP in a transgender individual. So you can see here the drug-drug interaction profile of various PrEP, considering 724 co-medication, basically all the co-medication that we have in the Liverpool website, except the HIV drugs, because we are looking at HIV prevention. And you can see that basically PrEP have really a low risk of drug-drug interaction. Just remind that you can use strong inducer with TDF, that the co-administration of TDF with nephrotoxic agent requires caution due to the risk of additional nephrotoxicity, that drugs that inhibit intestinal transporter can increase the absorption of TDF, which will result in higher tenofovir exposure, which could potentially increase the risk of renal toxicity. So you would have to monitor renal function when using TDF and these drugs. But also important to highlight that strong inducers are not possible with TAF and cabotegravir. So going back to the initial question of this case, and which is whether Adrian would be suitable to receive a long-acting cabotegravir as PrEP, well, the answer is probably yes, provided that St. John Wort, the strong inducer St. John Wort, is replaced by another antidepressant drugs with no inducing effect. Otherwise, there are no interaction with chemsex. You would have to think about the oral lead-in phase and not forget to separate the intake of oral cabotegravir with antacid. And there are no interaction with gender transitioning drugs. I think it's important to make this point very clear because interaction with hormone is really a big concern for transgender individuals, and which would actually prevent them from taking the PrEP or even HIV treatment, as it was demonstrated in some previous study. So in conclusion, cabotegravir, long-acting and other oral PrEP are characterized by a low risk for drug-drug interaction, and we are awaiting the result of the phase 3 study, which will provide a better understanding in regards to cabotegravir exposure response relationship for prevention in both men and female, and which will also provide a better understanding of the pharmacokinetic of cabotegravir in a special population. And with that, I would like to thank my colleague, and I would like to thank you for your attention. Thank you.
Thank you very much, Catia. That is a perfect introduction to our roundtable discussion. That was a great case. So I'll invite actually all our speakers to turn on their video and their audio. And we have a time for a few questions. We'll try to address some of the concerns and questions from the audience. So maybe we'll start off, Steve, some questions about long-term toxicities, potential toxicities of some of these cure, considering that these patients, many of them are not in a dire need. How does the conversation go with a patient coming in and asking about enrolling in one of these studies?
That's a great question, actually. It's actually interesting what's happening. So, you know, the big unmet need for cure is basically people not doing great on ART. And there's a lot of reasons for that around the world. But the people lining up to do cure studies are people doing well on ART. So, you know, and the reason why is there are a lot of reasons for that, but for the most part, for these types of studies, they're early, the phase 1, they're pretty risky. We want people who are healthy, and we want people with the virus under control. You know, I have never seen so much, what's the word? Oh, why am I blanking? People are doing this for the good, for just the help and to give back. We have particularly kind of the older generation of people who survived the 80s and the 90s. They're highly motivated. Altruism. If you ask people why they're doing it, we're enrolling people who are able to give really good informed consent, who know everything about the toxicities, and they know that they're not going to get cured, and yet they do this with some risk, and it's pure altruism. Because they know that this is what happened in the 80s and 90s. People took crappy drugs for a while, and eventually we found something that worked. And once we had something that worked, we built on it, right? And here we go today with the long-acting drugs. So that's how it works out. It's actually been very inspiring working in the cure environment. It reminds me of what was happening 20 years ago.
I think that makes a lot of sense. So Linda and Rick, we're going to jump to the future, and we just have a vaccine which is 70% protective. And now there's someone who's on PrEP, doing fine, and wants to get the vaccine. So do they, 70% protective, they get the vaccine, so do they continue the PrEP? Do they stop PrEP but have sex 30% less? I mean, what will actually happen in the real world with when we get these vaccines? Linda, you want to
So
head
Rick,
off?
yeah, I'll jump in first here. This sounds like a nightmare conversation, but I mean, I think so. So let me start by saying I think what opens up for us is this unbelievable opportunity to invoke choice amongst people. And it does mean that as clinicians, as providers, we're going to have to develop the art of having a conversation with our patients, with the clients that come to us. And I can't get away from the sort of the analogy of contraception for prevention. I think there's some fantastic lessons to be learned. I do work in Southern Africa where the HIV epidemic is largely among young women. So that analogy really does work for me. And, I would say that you'd have to have that conversation with this person. If they are ready to try a vaccine that is only 70% and then, go on that, that's going to have to be their choice. But we're going to have that conversation with them that if they wanted the belts and braces, the safety seat belt, then maybe they would continue PrEP for a time as well. I think we're going to have to feel our way in this environment. We've known, for example, for a long time, circumcision for cisgendered men is 60% effective. And we've had to obviously counsel men in a way that's a kind of vaccine, but it's not 100% effective. So, we've counseled men very effectively with that. And I think that'll probably be the uncomfortable position we're going to find ourselves in. But it's a whole lot better than when we only had ABC, I would say. I'm much happier with the problem we're going to have in the future than only having abstinence, be faithful, and condomize.
Yes.
I don't know, Rick, what do you think?
Well, 70% is great for the efforts and academic and intellectual contributions, but it doesn't cut it for someone who's having sex on a regular basis. A 1 out of 3 chance of failure doesn't sound like a workable deal to me. So as much as I feel strongly what you're saying, Linda, about choices, I think that's critical here, that we not dictate to patients what they should do and that they have a menu of options they can choose from. If a patient said to me, would I do a vaccine that's 70% effective? It really depends how frequently I'm having unprotected sex. And if I'm having unprotected sex once or twice a week, then I wouldn't do a vaccine that's 70% when I can choose something else that's effective 99%.
Well, Rick, maybe there the option would be, could you go with a coitally dependent option? So is that the time for a 2-1-1, so you get the background core protection, and you add the addition when there is that planned sex, and you're covered for those times when you don't have planned sex. I think it's going to be interesting to see how it plays out over time.
I think it'll make patients feel better to have both at their availability. So we'll do a lot that way. I just don't know. I just don't know whether people will say, well, that's good to have 2. I would rather have 1 that I can count on.
Mm-hmm.
Now, do you think, Linda, there's any chance that as we get more people who should be on PrEP, it'll be harder to do vaccine studies?
No doubt. And I think this is where the whole world is scratching its heads now and also going to the regulators to say, first of all, can we design, studies in a different way? This notion that we're going to have to enroll 40, 50,000 people to find that tiny additional effect or the non-inferiority sort of margin, I think is, first of all, is unaffordable. Also, where we're going to find all these people, how are we going to do this? So I do think it absolutely is a crucial time when we have to think, how do we design these studies better going forward? And, and maybe it does need, Mosaico is the first one that is interrogating this notion of choice. So people who don't see themselves, going down the oral PrEP route, who for reasons of whatever have failed or can't do oral daily PrEP, may then want to get themselves into a vaccine trial. So you're already kind of reducing the funnel. That would be one option. But otherwise, you know, I think we are going to have to think about how we do these trials in the future. And there's no doubt it gets more and more difficult. It's a, you know, it's a problem of our own success in a way. And yeah, I think that is something we just are going to have to work through over the next few years. But again, a good problem to have in many ways.
I want to add one quick thing, Jonathan. I think there's great interest for patients to get off of oral agents, though. And I think that fuels Steve's efforts to find cures. It fuels the injectable agents. And there's great fatigue among taking pills. And we don't know if that fatigue will diminish when it's a pill once a week or once a month. But I think the idea of an injection, because of the stigmatization and less frequency, will always have appeal no matter what success we have with the other agents.
Yeah, so actually that leads
Well,
Go ahead, Linda.
So, Jonathan, I was just going to say, and you know, in this part of the world, we know this, hormonal injectables are by far the way that people
Sure.
go in terms of contraception.
So along those lines, Catia, you know, David mentioned and Rick mentioned as well some of these cool new technologies, which are not only injections, these implantables and all sorts. Just a few words on those as we're moving. We may have options aside from intramuscular injections. What's been done so far in HIV? A few sort of examples that have been successful recently. I think you're muted, Catia. You have to unmute. Great, thank you.
Can you hear me?
Yes.
Well, I think definitively the possibility of having implantable option will be a great advantage. I'm thinking about, you know, if you have an inaugural disease and you have to treat someone with a potent inducer, so being able to, you know, remove your implantable drug will certainly be an advantage. And I mean, I know there have been some recent studies about TAF as implantable, which shows to be very promising. So I think as we move forward, there will be certainly more options. And as I have seen before, I think the drug-drug interaction does not seem to be a big issue with agent use as PrEP. We'll need to know a bit more about special population, because this is where I see a bit the problem for injectable. We need to really acquire this data. And we know that there is a large variability. We will have to learn about how to manage drug-drug interaction with injectable when you cannot, you know, stop the drug right away. That will be a bit the issue, and special population, yeah.
Great. Well, I think we're running out of time. A lot of additional questions coming in. We'll probably have to finish the roundtable. I want to thank all of you. This was a fantastic session. Thanks for the great talks and for the roundtable. And I'll move to the summary of our program. Thank you very much.
Thank you.
So in closing, I want to thank to thank all our speakers. Yesterday and today, we really had a spectacular lineup. I enjoyed all the talks. I think we had both great talks and great discussion. And I want to thank all of you who submitted questions. I'm sorry we couldn't get to all of them. We tried to deal with some of them, but definitely stimulating discussions. A special thank you to ViiV Healthcare, an independent educational grant, which was kind of them and allowed us to complete the program. Reminder of CME credits. You can claim your CME credits, which are good in Europe and can be transferred also to the United States. You can do so online. For those interested in a certificate of attendance, you can email us and you can get one provided. And most importantly, enduring material are available. You can go to this website at Academic Medical Education and actually download all this material. Next slide, please. For those interested, there is social media that Virology Education provides. You can see here LinkedIn, Twitter, and Facebook. You're welcome to be in touch. And give a shout out to our COVID-19 online educational program. We've been doing this now for a number of months with Dan Karitsky's and Anton Pozniak. Our next one will be on July 14th. And finally, let me ask that you complete the survey after the program. I want to thank you all for joining us, and good night.